what will happen? There is an enzyme called signal peptidase that will remove the signal peptide. So the signal peptide, or the leader peptide that was there, that was keeping it unfolded, is now cleaved off once it is released, when it comes out from the membrane. So the limitation of this transport system is that your folded proteins cannot be transported by this pathway. They have to stay unfolded in nascent form to be able to transport it across by this way. And once they are on the other side of the membrane, then they will start to fold and come into the active form. So here are some examples of, again, your target bacterium is E. coli. So we always have a lot of information on your E. coli and what are some of these leader peptides that are found in these exported proteins in E. coli. So when we compare these different leader peptides, there are certain commonalities that we found. These are some of the ideas that are common in all these proteins, at least about 7 or so of the proteins that have been studied in either in E. coli, that they have been found. So definitely all these proteins are from E. coli. Their leader peptide has 1, 2, 3 positively charged amino acids. This will be the end domain of it, which binds to the cytoplasmic side of the cytoplasmic membrane. And then after this charged amino acids, there is a string of 10 to 15 amino acids that are hydrophobic, have hydrophobic region, which we will call the H domain, which inserts through the cytoplasmic membrane. And finally, the 3 to 7 uncharged amino acids, which will be the C domain, which are found. So you see these commonalities in here. So if you look at these, the first one is the outer membrane lipoprotein. Now you see these lines here. These are boundaries between the hydrophobic and the hydrophilic areas of this protein. Whilst this delta sign tells you this is where the leader will be cleaved off. So in each of these, you can see those straight line to tell you the hydrophobic versus the hydrophilic. And then you can see where the cleaving will occur. When it emerges out of the membrane, the leader will be cleaved off, right? So this is the cleavage point, and this is the distinction between the hydrophobic and the hydrophilic areas. So some of the proteins are the lipoprotein, intermembrane lipoprotein in E. coli. The lambda receptor, what is lambda? Have you heard of lambda before? Anybody? You never heard of a lambda phage, bacteriophage lambda? It is a bacteriophage, it's a virus. So the receptor for lambda in the outer membrane, this is the protein, the leader peptide being shown of it. The periplasmic maltose binding protein. So this, where will you find this protein? This will be in the periplasm, where the maltose will be bound. So this is a periplasmic protein, and we are showing you the leader of it. Arabinose binding protein, which is also found in the periplasm. Then cytoplasmic membrane protein, where this is the FD. This is also a bacteriophage. Major coat protein is in the periplasmic space, and this is in the cytoplasmic membrane, with the minor coat proteins are found. So just to give you an idea that leader peptides are essential for transport through this transport mechanism, which we call, if we go back, which we call the SEC-dependent pathway. The GSP pathway will require the protein to be unfolded, and in the nascent way, they will be transported across, and the leader will be then cleaved off, and the protein folding will occur there. So here are some models of protein translocation, again shown in E. coli using the SEC-A and B idea. It's a simple kind of a model for you to look at. So your SEC-B is taking the protein, pre-protein with this dark band is of course your leader peptide that is there. The SEC-B brings it to SEC-A, which is bound to the membrane. At that point, SEC-B will be released. Now the protein is attached to the ATPase, and then the leader gets inserted by a flipping over, where you can see the leader going across the membrane, and this protein being pulled through so that it goes across. This is just a model. Uh, and then the ATP breakdown occurs, and your protein is released out. Here is another model where here the translocation, and the final part of it is done by delta P, and of course delta P is the potential energy in the electrochemical proton gradient, which is also needed along with the ATP hydrolysis. And in this one, where there is, in the absence of delta P, you will need several rounds of SEC-A and ATP hydrolysis. So you can see SEC-A being involved more than one time, and ATP being hydrolyzed and transporting this protein if there is no delta P and the electrochemical proton gradient has been diffused away. Here is a model for a SEC-dependent translocation of membrane proteins. It's not being secreted, but it is being embedded in the membrane only. So those proteins that get embedded in the cytoplasmic membrane, here is some models of it where you will be needing the mechanism where there is the leader again, and the leader allowing. So here is the periplasm, and here's the cytoplasm, and the membrane being put in, membrane protein being embedded into the cell, into the membrane as such. Okay, so these are some models for other membrane proteins. Now, going away from this SEC-dependent pathway, is there other SEC pathways also which are similar, secretion pathways that are similar? So we just discussed a SEC-B pathway, which uses the chaperone as SEC-B. While there's another pathway that is operative in some bacteria, which is called the SRP pathway. So SRP is again a secretory system, and this is mediated by a complex of RNA and protein. So it's not only the protein SEC-B, but another protein RNA complex guides this protein to be transported across. So SRP stands for signal recognition particle. This is a complex of RNA and protein together. The signal sequence is recognized by the SRP, and it directs the protein bound to the ribosome to the SEC-YEG. So only this difference is that we use not SEC-B as the protein that guides it, but the SRP, which is actually has an RNA in it also. Okay. Both pathways use the SEC-A, which is an ATPase for translocation, and during translocation, signal peptidase cleavage and at the amino terminal, the N terminal will occur. So the rest of it is same, just that there is no SEC-B here, but this is the SRP, okay? Now, to tell you that within the same bacterium, the gram-negative bacterium, they use the SEC-B pathway to transport outer membrane proteins, but the cytoplasmic membrane proteins are embedded using the SRP pathway. So the same bacteria may use both pathways to transport different proteins, either if it's going to the outer membrane protein or it is a cytoplasmic membrane protein. Two different pathways are being used. So what I'm trying to say is it's not exclusively that one bacterium will use only SEC-B and not use SRP, but even within the same cell, different pathways can be used to take the protein up to the cytoplasmic membrane or take the protein up to the outer membrane. In gram-positives also, such GSP mechanisms have been noted, but the SEC-B has not been identified. Similar mechanisms are there. So to compare and give you a visual of what we are talking about, here is the model for SEC-B transport, and here is the model for SRP transport. I'm going to move this up again. You can see that the ribosome is producing your protein, the nascent protein. The SEC-B attached to it, there is of course the signal also, leader peptide there. SEC-B binds to it so it doesn't fold anymore also, and it transports it to SEC-A, where the SYEG complex has made a pore for it, and through here the protein will be allowed to go into the periplasmic space, and in the periplasmic space it is kept unfolded, again unfolded by certain other different proteins for different mechanisms for different proteins there. And then it will be directed towards the outer membrane protein outer membrane where the protein will be embedded, embedded in the outer membrane. So here's the other mechanism where the ribosome is producing the nascent protein. The SRP is the one that is going to allow it to be transported. But actually, the SRP will take the whole ribosome with it up to the sec-A and allow the protein to get transported across the YEG complex. And you are actually embedding this cytoplasmic membrane protein using the SRP mechanism. Here, the SEC mechanism was being used to insert the protein in the outer membranes. It becomes an outer membrane protein here, and it becomes a cytoplasmic membrane protein here. You can see that this mechanism cannot be used for outer membrane proteins because the ribosomes will not be found in the periplasmic space. So most of the cytoplasmic membrane proteins may be transported and translocated by this SRP mechanism, whilst the outer membrane proteins will be embedded in the outer membrane using the SEC-B mechanism. Moving on. The next system that we will discuss is the TAT system. This is a protein translocation system found in bacteria and some archaea, where it allows the protein to be moved across the plasma membrane. But the idea is that it transports folded proteins. So the TAT pathway translocates folded proteins. But the important thing is that they also have a signal sequence. And in the signal sequence, you will have twin arginine residues. So that's why it is called the TAT, twin arginine transport system, right? So the signal will have the twin arginine residues in it. And an example of it is an enzyme that is found in E. coli, which is, of course, a cytoplasmic membrane protein, the enzyme called formate dehydrogenase. This hydrogenase in E. coli, this hydrogenase is found as a cytoplasmic membrane protein, and it is embedded in the membrane using the TAT pathway. If it is a gram-negative bacterium, of course, there has to be an outer membrane transport system also. Usually, the TAT only works with the type 2 secretion system in the outer membrane. Okay, so TAT works with type 2 secretion system, and I'll tell you what the type 2 secretion system in a little bit. So again, we talk about these secretion systems. So 6 to 8 different secretion systems have been identified in gram-negatives. So gram-negatives definitely have more because of this outer membrane that they have. Of these 6 to 8 that have been identified, and more have been identified more by now probably, 1 and the 4 types are also found in gram-positives. Gram-positives only have the cytoplasmic membrane, but the type 1 and 4 they look just about the same as the outer membrane transport systems. So 1 and 4 have been looked at in gram-positives also. Type 2, 3, and 5 are unique to gram-negatives. These will only be found in gram-negatives. And most of these type 2, 3, and 5 are the ones that secrete virulence factors. You know, some of the gram-negatives release virulence factors. So definitely these will be used mainly for secreting virulence factors from gram-negative bacteria. Type 1 pathway transports proteins across the outer membrane that were first translocated across the membrane by the SEC-dependent pathway. So type 1 works with the SEC pathway. Type 3 is a SEC-independent pathway where SEC is not used, but they form injectosomes, and we'll look at this very soon. And it also transports, of course, as we said, that virulence factors are released by these. So type 3 transports virulence factors and other proteins. Type 5 are SEC-dependent. So every one of them has a different condition that you have to learn about. And an important one is the autotransporter here, where it transports itself. You will be reading, the next reading will be on these protein secretion mechanisms and how they were deciphered. So the next papers that the students will be presenting one of them will be on autotransporters. And this is a very interesting paper. Actually, it is a review which will help you understand how these transporter proteins were found at different in different microbes. Moving on. So protein translocation across the outer membrane as such is what we will talk about, and we will discuss all these type 1, 2, 3, 4, 5, and so on. So proteins crossing the cytoplasmic membrane by the general secretion pathway, they are now in the periplasmic space. So they will use type 2, 4, and 5, and also another pathway which will be called chaperone-usher pathway. Sec proteins are not involved in type 1 and 3. So again, the same, the delineation of which one matches with what. And as we go, we will give you examples of all of this. Type 1 is similar to the ABC transport system, which is found in your cytoplasmic membrane. Type 3 is, of course, where the syringe will be formed. And you will see a commonality between the type 3 and your flagellin proteins being exported to make the filament of the flagella. So you will see type 3 talked about when we talk about the flagellin protein being transported to make the filament of the flagella. So, so model for biogenesis of pili. The pili that are formed, again, here the pili will use the chaperone-usher pathway. And this is the cell surface protein, which is, of course, a protein that is used for virulence, such as the pilus production will use this pathway. And in this one, no energy is needed. The protein in the outer membrane protein called the usher is used to transport the pillin protein. So if you're looking at it, we have the pillin proteins that will make the pillar being made in the cytoplasm. And using the Sec pathway, unfolded protein is going through the Sec pathway, where in the periplasm, the Sec will be, the leader will be chopped off. And here you will see that another protein, which is the D protein, assisting it to take it to the usher, taking it to the channel through which the outer membrane, through which this protein will form the pilus. So the pilus formation uses this chaperone-usher pathway. So this is your chaperone and this is your usher. The PAP-D is the chaperone, and definitely the C is, of course, the channel, which will be the usher. So we call this the usher, we call it the chaperone. So the chaperone takes it from the cytoplasmic membrane up to the usher, and through which these proteins will be creating a pilus formation. Now remember, pilus is also a outer, appendage, which not only grows, but it can be retracted. We talk about it when we talk about conjugation. When the cells are going to conjugate, pilus makes the attachment for the donor to the recipient, but then the pilus is retracted so that the recipient comes in close contact, and then the DNA transfer occurs. So pilus formation always involves not only production of the pilus or the retraction, and the retraction of the pilus will also be discussed later. But anyway, in this case, chaperone-usher pathway, the example for a chaperone-usher pathway is the production of the pilus in your gram-negative bacteria, such as E. coli. So again, so also the type 1, which is the outer membrane transport protein transporter, is very similar to the ABC. So we will compare type 1 with ABC. So this type, it's also called the type 1 protein secretion pathway. This is found in prokaryotes, quite common. And it transports proteins from cytoplasm across both plasma membrane and the outer membrane. So it is actually a combination where both this mechanism will transport proteins across both the membranes. In this case, if the protein is a secreted protein, it will also have another signal, which is called the secretion signal in their C-terminal. So remember, the leader signal is in the N-terminal end of the protein, while the secretion signal is in the C terminal of the protein. Gram-positive bacteria use a modified version of this pathway to translocate proteins across the plasma membrane. So ABC type 1, as we call it, remember they have a terminal secretion sequence also. So here is a model given to you to understand how this is happening. So proteins are being transported by the ABC pathway. These do not have an N-terminal signal, but they have a C-terminal sequence, secretion sequence. In this case, the channel is formed through the two membranes directly. Okay? There's a membrane fusion protein that connects the outer membrane proteins to the ABC system in your cytoplasmic membrane. So the protein is being brought in through the ABC system, and across the membrane fusion protein, they will go. And in the outer membrane protein, they will go across that. The channel is formed through and through. And actually, the C-terminal sequence is there, which will allow the protein to be secreted out. Otherwise, this protein would not be able to be secreted. So going across the outer membrane, in this type 1, you need the C-terminal secretion sequence there. Talking about the second one, which is your type 2. Type 2 uses SEC, again, the secretory system, the general secretory pathway, to cross across the cytoplasmic membrane, and then folds in the periplasmic space. But then after that, the folded protein goes through the outer membrane system, which is the GSP-D and S system. So again, here is your GSP-D and GSP-S. Again, this is, of course, an ATPase here. So definitely, this will be as a folded protein going across the outer membrane. Okay. So this is usually the GSP-D, the secretin. This is a complex of GSP-S lipoprotein that serves as a gated channel for secretion of substrates to the cell surface. Okay, GSP-C, of course, is your the channel. If it is going through this system, the whole GSP system, and not using the SEC system, then folded protein will go through the GSP-C and then GSP-D and will be secreted out. So folded proteins will go this way. Unfolded proteins may go through the SEC system, get folded in the periplasm, and then also use the same channel to be released out of the outer membrane. I think we've discussed enough of them for today. I think you might be quite saturated with all this much information. I'll give you the time to think over it and look through this in your book, as well as the slides that I have presented, and see